Next up, we have Dr. Lynn Hudson, who serves as the Chief Science Officer for the Clinical Path Institute and is the Executive Director of the International Neonatal Consortium and the Multiple Sclerosis Outcome Assessments Consortium. She oversees the matrix of regulatory science initiatives for CPATH Consortia and co-chairs the Scientific Advisory Committee of CFAST, the partnership between CPATH and CDISC for creating and maintaining therapeutic area data standards.
So this morning we heard from Janet Woodcock, who was one of the chief architects of the Critical Path Initiative, which was a whole list of all sorts of opportunities that could be undertaken to try to accelerate medical product development. And one of those solutions was to form public-private partnerships, to get together all the right expertise, to share data, expertise, and de-risk drug development in that process. Janet also was behind the founding of the Critical Path Institute. And so what I'm going to try to do is go through some of the examples of the drug development tools that we work on through the Critical Path Institute, also known as CPATH, and how we have drawn on NIH work and how NIH can help with this whole process. So we have a number of consortia in CPATH. Some of them are disease-specific, like for Alzheimer's, Parkinson's, multiple sclerosis. Some are cross-cutting consortium that work on drug toxicities. So our predictive safety testing consortium is one of those examples. And I will go through a couple of our qualifications that illustrate how you can speed up drug development. So for example, we have 3 qualified patient-reported outcome instruments. So for the instrument on the non-small cell lung cancer symptom assessment questionnaire, now all the companies that are developing drugs for that particular lung cancer can use this one PRO instrument as a primary or secondary endpoint. And when they send their data into FDA, FDA doesn't have to both evaluate the instrument itself and the data that was produced with that instrument. So instead of, it shortens the FDA review time rather than having 10 companies bring in their home-brewed PRO instrument with their data to have one instrument that everybody can rely on. Data is central to everything we do at CPATH to develop any of these drug development tools. The culture of data sharing has changed dramatically in the past decade, and we get a lot of individual patient-level data, not only from our industry members, but also from NIH-funded studies. What we do with data is that we have to standardize it, and that's another objective that FDA has implemented that has really sped their ability to review applications. So a couple years ago, FDA required that all submissions be in CDISC data standard format. And we've actually helped develop about 40 different data standards that are now used for individual studies. And what we do when we get data is that we map all the data to a standard, and then we are able to pool that data together. And that data is used for our Alzheimer's database. It's been used all over the world for really a whole range of different research questions. And it's been very helpful for different researchers, but what we used it for was to come up with a disease progression model across the entire Alzheimer's disease continuum that also allowed us to develop a clinical trial simulation tool to better design clinical trials. So once you know a few covariates, like the results of memory tests or the ApoE4 genotype, you can start putting those covariates into a model, and that really helps you design your trial. Another approach that we've done with our multiple sclerosis consortium is to take and examine performance measures to see what we could use as primary or secondary endpoints in trials. And for that, we relied on a lot of data that was originally accumulated in academic studies. So these different performance measures, like a timed 25-foot walk for walking speed, were used not just for multiple sclerosis, but the National MS Society convinced all of the sponsors in the field to incorporate those tests into their trials and in that way validate their performance. So see what a difference in walking speed, a 4-second difference in walking a timed 25-foot walk would mean to a person to MS when it comes to something very practical, like whether they could cross the street in time before the walking light goes off. Oops. And then another example is for polycystic kidney disease. Before our consortium started, there wasn't much activity at all in doing clinical trials in this space because there were no good endpoints. There was no good ways to measure as your kidneys expand, and finally the kidneys fail. So you needed to have endpoints that would make sponsors actually be able to see if their drug was giving a beneficial effect. So we used total kidney volume. We got it qualified as a prognostic biomarker, and now it's being used as a reasonably likely surrogate marker, so an endpoint. And once that was qualified, more than half a dozen companies are now in the space of this, trying to develop different candidates. We have some consortium. The neonatal is a good example of where there is no data. There is no data because it's really hard to do clinical trials with neonates. The last drug approved for neonates was in 1991, surfactant. And almost all of the drugs used in neonatal intensive care units are used off-label. So to start for a consortium like that, it's just trying to figure out what data you need to collect, how to collect it, and do some kind of basic things like trying to do what Janet was referring to, getting together master protocols that could be used in platform trials for neonatal trials. So this gets to the gaps in knowledge, the huge gaps of knowledge we have in drug development, and how NIH can help fill them. And I should start by saying that NIH studies have already filled a lot of these gaps, but there's a lot more that can be done, and I just gave you an example with neonates. There's a huge need for just the natural history studies. So what's normal blood pressure in a neonate? What's a normal lab value? There's a need for really understanding the underlying pathology. So many clinical conditions, they may look the same, but they have very different disease underlying. So we need more robust biomarkers. NIH does a terrific job with the discovery of biomarkers, and we need to follow it through with validating those biomarkers in clinical trials. NIH can also do a lot for collecting and analyzing real-world data because there's going to be populations, and rare disease is one of those, where most of the data that you're going to get in is not going to be clinical trial data. It's going to be real-world data from the use of drugs in those populations. And then finally, an area where, again, Janet touched on as the need for improved and innovative clinical trial designs, that's an area where the best modelers in the world are really in the industry. So it's, but NIH can still contribute to that, trying to improve the methodology for clinical trials. And I think that I will stop here with only I'd like to just say that sometimes, since the tagline for NIH is turning discovery into health, sometimes in the NIH portfolio, it's more exciting to fund an intervention trial than it may be to fund some of the basic infrastructure that you'd want before you start an intervention trial. So, for example, again, for neonates, the only hard endpoint you have is death, because there haven't been good endpoints developed. So it may sometimes be more worth investing in those basic kinds of strategies to better design a clinical trial than embarking on a clinical trial. So I'll stop there. I'm done.